Whole blood transcriptomic signature of Cushing syndrome by Birtolo et al. Cushing syndrome is a condition characterized by chronic cortisol excess due to glucocorticoid treatment or endogenous hypercortisolism. Currently, cortisol assays are routinely adopted as part of the regimen for diagnosing Cushing syndrome. There is no biomarker available to directly measure the biological effects of glucocorticoids at the tissue level and the individual consequences of glucocorticoid excess. The aim of the study was to identify biomarkers able to better quantify the individual biological impact of glucocorticoid excess through the analysis of whole blood transcriptome. 57 whole blood samples were taken from patients with Cushing's disease and patients with benign adrenal Cushing syndrome. The samples were further classified into 4 categories, including overt Cushing syndrome, mild Cushing syndrome, eucortisolism, and adrenal insufficiency. The RNA was sequenced on the platform Illumina, and the data was analysed via PCA and LIMA. PCA showed that the first principal component was enriched with genes related to immune response pathways, especially neutrophil activation. Many genes were identified from the study, including the gene FKBP5. The samples were split into training and validation cohorts to predict glucocorticoid status. The transcriptome predictor was optimized in the training cohort with the 1500 most variable genes to detect overt Cushing syndrome. It was found that FKBP5 gene expression alone predicted overt Cushing syndrome with an accuracy of 0.76. Firstly, whole blood transcriptome signature reflects the biological action of glucocorticoid excess. Secondly, FKBP5 expression could be a non-hormonal gene marker for Cushing syndrome. Finally, the clinical relevance of the study warrants further investigation in larger patient groups, mainly with moderate Cushing syndrome. For further information, please scan the QR code.